Today's topic is cerebrospinal fluid analysis, that is CSF analysis. CSF actually it is a clear and colorless fluid formed within the cavities of brain ventricles and around the spinal cord. It is formed from choroid plexus and returns to the blood vessels in the lumbar system. It is actually a transudate of plasma. You should know the difference between transudate and exudate. Here the volume of CSF is 125 to 150 ml per day. The function is it acts as a shock absorber and it regulates the intracranial tension. Here the collection of cerebrospinal fluid procedure name is spinal puncture in the lumbar area between L3 and L4 and L4 and L5. We have the normal and various disorders of the CSF fluid analysis that called composition or biochemical composition of CSF. We have normal some diseases, that is brain tumor, subarachnoid hemorrhage, viral infection, TB, meningitis, and bacterial purulent meningitis. You have to mention about the color and appearance, glucose content, and protein content. Usually the glucose content in CSF is 50 to 80 milligram per day per DL, whereas protein content is 20 to 40 milligram per DL. So in brain tumor, glucose is decreased whereas protein is increased. In subarachnoid hemorrhage, the glucose is normal whereas protein is more than 40, that is increased. TB meningitis, the important point is slight yellow and opalescent color, the cell count is increased, glucose is relatively low, and protein is increased. The coagulation picture of tuberculous meningitis is very, very important. Cobb web type. Bacterial opalescent, decreased glucose, increased protein. Bye.